crowning glory of the central nervous system is, of course, the brain. And anatomically, the brain is the part of the central nervous system that fills the upper portion of the skull. So I thought it would be prudent to bring out my skull, which was a gift to me from a friend of mine who was a physician who passed away many years ago. And this is a human skull, and if I could show you the bottom of the skull, you could see here where the brain stem and the central nervous system would connect through the spine. And if I open up the top of the skull for you, you will be able to see how the brain would fit inside this particular skull. Although it only weighs about 1.5 kilograms and could be held in one hand, the brain contains billions of interacting cells that integrate information from inside and outside the body and coordinate the body's actions and enable humans to talk and think and remember and plan and create and dream. And because of its central importance for behavior, the brain is the subject of the next 3 sections of our chapter. Those who conduct research on the brain and other parts of the nervous system are called neuroscientists. And brain research involves collaboration by neuroscientists with other disciplines, including anatomy, physiology, biology, pharmacology, neurology, neurosurgery, psychiatry, and psychology. To discover how parts of the brain are related to behavior, neuroscientists are faced with formidable tasks, and mapping out the brain functions requires a working brain. Thus, special methods are needed to discover the relationship between brain activity and behavior. Some methods include examining changes in behavior that accompany a brain damage resulting from tumors or strokes or head injuries or other misfortunes. And this is case study methodology. Doing research with patients with brain damage has its limitations because subjects are not plentiful and neuroscientists can't control the location or severity of patients' brain damage. Neuroscientists sometimes observe what happens to brain structures in animals that are purposely disabled. Lesioning, for example, involves destroying a piece of the brain. Electrical stimulation of the brain, or ESB, is where a weak electrical current is sent into a brain structure to stimulate or activate it. The EEG, or the electroencephalograph, is a device where recording electrodes are attached to the surface of the scalp, and that permits the EEG to record electrical activity in the cortex over time. The EEG provides output in the form of line tracing called brain waves, and brain waves vary in frequency and amplitude, both in cycles per second and measured in voltage. Various states of consciousness are associated with different brain waves, and characteristic EEG patterns for alert wakefulness, drowsiness, and deep dreamless sleep are shown here. The use of the EEG in research is discussed more in Chapter 5. Transcranial magnetic stimulation is a technique that permits scientists to temporarily enhance or depress activity in a specific area of the brain. And in this particular situation, the TMS, a magnetic coil, is mounted on a small paddle and is held over the specific area of a subject's head. And the coil creates a magnetic field that penetrates to a depth of about 2 centimeters, and by varying the time and duration of the magnetic pulses, the researcher can either increase or decrease the excitability of neurons in the local tissue. This particular technology allows scientists to create virtual lesions in humans for short periods of time using a painless, non-intrusive method. The CT scan is used to examine aspects of brain structures, and they can provide enhanced x-rays of horizontal slices of the brain. where an X-ray beam and an X-ray detector rotate around the patient's head, taking multiple X-rays of a horizontal slice of the patient's brain, and B, where a computer combines X-rays to create an image of a horizontal slice of the brain. And this can show a tumor, so in blue there you can see the tumor on the right. MRI scans can be used to produce remarkably high-resolution pictures of the brain, and a vertical view of the brain from the left side is shown here. Like PET scans, functional MRIs can monitor chemical activity in the brain, and this image shows regions of the brains that are activated by a flashing light. PET scans are used to map brain activity rather than brain structure, and they provide color-coded maps that show levels of activity in different areas of the brain over time. The PET scan shown here pinpointed 2 areas of high activity indicated by red and green colors when a research participant worked on a verbal short-term memory task. Now that we've reviewed the selected neuroscience techniques, let's examine what researchers have discovered about functions of various parts of the brain. And the brain can be divided into 3 major regions: the hindbrain, the midbrain, and the forebrain. And the principal structures found in each of these regions are listed here. And so let's take a quick look at these. The hindbrain includes the cerebellum and 2 structures found in the lower part of the brainstem called the medulla and the pons. The hindbrain is located at the lower end of the brain where the spinal cord joins the brainstem. The medulla is in charge of circulation, breathing, muscle tone, and regulating reflexes. The pons is important in sleep and arousal. The cerebellum is critical in the coordination of movement and equilibrium. The midbrain is the segment of the brain stem located between the hindbrain and the forebrain. The midbrain lies between the two, and it involved in sensory functions such as locating where things are in space, and it also contains structures which are important for voluntary movement. The reticular activating system is found in both the hindbrain and the midbrain, and is important in sleep and in arousal, as well as breathing and pain perception. The forebrain is the largest and most complex region in the brain, encompassing a variety of structures, including the thalamus, the hypothalamus, the limbic system, and the cerebellum. The thalamus is the way station for all incoming sensory information before it is passed on to the appropriate higher brain regions. The hypothalamus is the regulator of basic biological needs such as hunger, thirst, sex drive, and temperature regulation. The limbic system is a loosely connected network of structures that are involved in emotion, motivation, memory, and other aspects of behavior. The cerebrum is the largest and most complex portion of the human brain, and the convoluted outer layer of the cerebrum is the cerebral cortex. The cerebrum is responsible for complex mental activities such as learning, remembering, thinking, and consciousness. The cerebrum is divided into 2 specialized hemispheres that are connected by the corpus callosum. The corpus callosum is a thick band of fibers that transmits information between the hemispheres, and researchers using split brain methodology have learned that each hemisphere is specialized for different functions, with left usually dominant for language and right for spatial skills. The occipital lobe, where the primary visual cortex is located, the parietal lobe, where the primary somatosensory cortex is located, the temporal lobe, where the primary auditory cortex is located, and the frontal lobe, where the primary motor cortex and executive control system is located in the brain. In split brain surgery, the bundle of fibers called the corpus callosum, which connects the two hemispheres, is severed in order to decrease the seizures from epilepsy. Each hemisphere's primary connections are to the opposite side of the brain. There are perceptual asymmetries between the speed of auditory and visual processing of the left and the right hemisphere of the brain. The left hemisphere is better at tasks involving verbal processing like reading and writing, and the right hemisphere is better at non-verbal processing tasks such as spatial, musical, and facial recognition tasks, as well as emotional processing. Hormones are chemical messengers in the bloodstream that are secreted by the endocrine system or the endocrine glands in a pulsatile release by endocrine glands. This is several times per day in brief bursts and or pulses, and the level of many hormones are increased to a certain level, then signals are sent to the hypothalamus or other endocrine glands to stop secretion of that hormone, and that's a negative feedback system. These glands include the pituitary, the master gland that secretes substances influencing the operation of all other glands, as well as the growth hormone and oxytocin. The thyroid gland controls metabolic rate. The adrenal glands control salt and carbohydrate metabolism. The pancreas secretes insulin to control sugar metabolism. And the gonads secrete sex hormones involved in the development of secondary sex characteristics and reproduction. Questions of how much of behavior is biologically based and how much of behavior is environmentally based are very old ones in psychology. Since 1970, however, research methodologies have been developed in the field of behavioral genetics that shed new light on the old nature versus nurture question. Basic terminology necessary for the discussion of basic genetic research include chromosomes, which are strands of DNA carrying genetic information. Each human cell contains 23 or 46 total chromosomes, with the exception of sex cells, which have 23 single chromosomes that are not yet paired. Each chromosome contains thousands of genes, which also occur in pairs. Sometimes a member of a pair has a louder voice, always expressing itself and masking the other. Different member of the pair. This is a dominant gene. A recessive gene is one that is masked when the paired genes are different. When a person has two genes in a specific pair that are the same, the person is homozygous for that trait. If the genes are different, then they're heterozygous. Genotype refers to a person's genetic makeup, while phenotype refers to the way in which a person's genotype is manifested in observable characteristics. You can have different genotypes which yield the same phenotype. So, for example, one person has two genes for detached earlobes, which is dominant, while another has one for attached and one for detached. Both outwardly look the same in the earlobe department. Most human traits are not so simple with regard to genetic transmission. They are polygenic or influenced by more than one pair of genes. Investigating heredity includes various research methods. Family studies simply assess hereditary influence by examining blood relatives to see how much they resemble one another on a specific trait to determine whether it runs in the family. Many things run in families, however, that are not genetic, like dessert recipes or speaking a certain language. Twin studies can yield better evidence about the possible influence of heredity because identical twins have the exact same genotype. They share 100% of the same genes. Fraternal twins only share 50% of genetic relatedness, the same as any two siblings born to a set of parents at different times. Twins of both types, however, are raised in more similar environments, same age, configuration of relatives, and if identical twins are more similar on a given trait than fraternal, the assumption is that it is probably genetic. Adoption studies assess genetic influence by comparing adopted children with both their biological and adoptive parents. And if they are more like their biological parents, whom they have never met, on a trait, it is probably genetic. Genetic mapping is the process of determining the location and chemical sequence of specific genes on specific chromosomes. Clustered, regularly interspaced, short palindromic repeats store a small section of harmful viruses in order to recognize them when next attacked. Scientists can use these to modify the genomes of animals, including humans, in order to help prevent genetic diseases like cystic fibrosis or Alzheimer's. Epigenetics is the study of the heritable changes in gene expression that do not involve modifications to the DNA sequence. Modern advances in molecular genetic technology have allowed for genetic mapping, the locating of specific chromosomes, and genes involved in phenotypic expression. While this is a promising area of research, initial progress points toward the complexity of polygenic inheritance rather than yielding simple answers to the nature-nurture debate. The behavioral genetics field has yielded much the same conclusion. There'll be no simple answer to is it nature or nurture question. Richard Woe's quote says, "We inherit dispositions, not destinies." The field of evolutionary psychology is a major new field in psychology focusing on analyzing human behavior in terms of adaptive significance. It is based on the work of Charles Darwin and the ideas of natural selection and the reproductive fitness, i.e. the variations in reproductive success, are what really fuels evolutionary change. Evolutionary theorists study adaptations or inherited characteristics that increase in a population because they help solve a problem of survival or reproduction during the time they emerge. So, for example, giraffes and long necks may extend even when no longer needed. For example, the fight or flight response may have been very helpful in primitive times, but now it is related to a number of stress-related diseases. And similarly, humans show a taste preference for fatty foods. This was adaptive in a hunter-gatherer society when dietary fat was scarce, before potato chips. But now this preference results in obesity, heart disease, and while this may lead to decreased longevity, the effects of reproduction success is much more difficult to gauge. Parental investment refers to time, energy, survival risk, and foregone opportunities that each sex has to invest in order to produce and nurture offsprings. Trivers in 1972 suggests that a species' courting and mating patterns are based in parental investment. When parental investment is high for females and low for males, polygyny results. A mating system whereby each male seeks to mate with multiple females and each female seeks only one male. Polyandry occurs when each female seeks to mate with the multiple males and each male with only one female. This emerges when parental investment is high for males and low for females. So monogamy emerges when male and female parental investment is roughly be equal. A mutation is a spontaneous, heritable change in a piece of DNA that occurs in an individual organism.